Welcome back, everyone, for another deep dive. This time, we're headed into the fascinating world of molecular biology.
Oh, yeah.
But don't worry,
Oh, yeah.
no lab coats necessary for this one.
You won't need your pipettes.
We're going to be exploring how scientists separate and analyze those tiny but super crucial molecules.
They are tiny.
DNA, RNA, proteins.
Yeah, you got it.
It's going to be like getting a peek behind the curtain of all this cutting-edge research.
Exactly.
You know, maybe enough to impress your colleagues at your next meeting.
Oh, for sure.
Or just satisfy that burning curiosity you have about how science actually works.
Absolutely.
And for this deep dive, we'll be taking a close look at an article from Assay Genie,
Great article.
which compares two key players in the lab,
Yeah.
Agarose and polyacrylamide gels.
The gels.
Oh yeah, the gels.
This is a good one.
These aren't the gels you find in your skincare routine, though.
Not at all.
These gels are actually the foundation of a technique called gel electrophoresis, which is basically using an electric field to sort molecules based on their size, shape, and charge.
It's like a race.
It is kind of like a race.
Where everyone has different
Yeah, like everybody has,
size shoes, and some are even carrying weights.
yeah, different shoes. Maybe somebody's got like ankle weights on or something.
Exactly.
But let's start with the racetrack itself.
Okay.
What exactly are these gels made of?
Well, agarose, surprisingly enough, comes from seaweed.
Seaweed.
It's a natural polymer that forms this porous matrix,
Yeah.
kind of like a sponge when it cools down.
All right.
This makes it great for separating larger molecules like DNA fragments.
So I'm picturing like a microscopic obstacle course.
I like that.
Agarose is like a field of boulders,
Yeah.
tougher for those bigger molecules to navigate.
Yeah, that's a good way to visualize it.
Okay.
Now, on the other hand,
Right.
we have polyacrylamide,
Okay.
which is synthetic
Uh-huh.
and made by linking together molecules of acrylamide and bis-acrylamide. And this creates a much tighter network
Okay.
with smaller pores.
Got it.
Perfect for those smaller molecules like proteins or even tiny fragments of DNA or RNA.
So polyacrylamide is less of an obstacle course
Yeah.
and more like a tightrope walk for those tiny molecules.
Yeah, I like that.
Sounds like the choice of gel really depends on what you're trying to separate.
Precisely. Is that right?
Choosing between agarose and polyacrylamide is a bit like choosing the right tool for the job.
Sure.
You wouldn't use a hammer to tighten a screw, would you?
Definitely not.
Yeah.
But let's dig into how this separation actually works.
Okay.
The article mentions size-based separation as a primary principle.
Right.
I'm guessing this means larger molecules get slowed down more
Yeah.
because they have a harder time squeezing through those pores.
Exactly. Think about it like trying to navigate a crowded room.
Okay.
It's a lot easier if you're smaller and can weave through the crowd.
Makes sense.
Larger molecules encounter more resistance in the gel matrix and move slower as a result. Okay, that makes sense.
Yeah.
But the article also mentions something called molecular sieving.
Yes.
Does that have to do with size as well?
It does, but it takes size-based separation to a whole new level.
Okay.
It's not just separating big from small,
Oh.
but actually distinguishing between molecules that are very close in size.
Wow.
Polyacrylamide, with its incredibly fine pores, is the master of molecular sieving.
So it's like having a set of super precise filters
Yeah.
that can separate molecules based on like the tiniest differences in size.
Exactly.
That's incredible.
It is, and this level of precision is crucial for techniques like SDS page.
SDS page.
Yeah.
Okay.
Which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis.
Oh, wow.
I know that's a mouthful,
It is a mouthful.
but bear with me because this is where things get really exciting.
Okay, I'm all ears, especially if it involves acronyms that make my head spin.
All right.
But seriously, what is so special about SDS page?
Well, SDS page uses this molecular sieving phenomenon to determine the molecular weight of proteins.
Okay.
It essentially gives all the proteins a uniform negative charge,
Uh-huh.
so they all behave the same way in the electric field.
Gotcha.
Then they're separated purely based on their size.
So it's like leveling the playing field,
Yeah.
eliminating all those other factors like shape and their original charge,
Right.
and turning it into a pure size-based race for the proteins.
You got it.
Okay.
It allows scientists to determine the precise molecular weight of each protein in a sample.
That's incredible. It's like having a molecular weigh station
Exactly.
for proteins.
And knowing the molecular weight of a protein is like having a key piece of its identity card.
Okay.
It helps us understand its structure, function, and even how it might interact with other molecules.
Okay. Now that's what I call an aha moment.
Right.
We started with these gels from seaweed and synthetic polymers.
Yeah,
And now we're talking about identifying proteins and understanding how they work.
that's right.
It's amazing how these seemingly simple techniques
Yeah,
can lead to such groundbreaking discoveries.
it really is. And we've only just scratched the surface.
Oh, wow.
There's a whole world of applications for both agarose and polyacrylamide gels
Okay.
in various fields of research.
Well, I don't know about you,
Yeah,
but I'm ready to dive deeper into those applications.
me too.
We've got the basics down,
Uh-huh.
but I'm itching to see how these gels are actually used
Right.
in real-world research.
Let's do it.
Let's shift gears and explore some specific examples, shall we?
Let's go. All right, let's step into a genetics lab and see agarose gels in action.
Okay.
Imagine you're a researcher studying a gene
Uh-huh.
that's suspected to be linked to a specific disease.
Okay.
You've collected DNA samples
Right.
from both healthy individuals and those with the disease.
Okay. I'm picturing rows of test tubes, DNA strands swirling inside.
All right, so what's next?
Yeah, what's next?
Well, you'd use a technique called PCR
PCR.
to amplify that specific gene, creating millions of copies.
Wow.
So you have enough material to analyze.
Okay.
Then comes the agarose gel.
Right.
You load those amplified DNA samples into the gel
Okay.
and apply an electric current.
So this is where that molecular race really gets going.
Right.
The DNA with its negative charge will be drawn towards the positive electrode,
Exactly.
and the smaller fragments facing less resistance will sprint ahead.
Precisely after a while,
Okay.
you'll see these distinct bands forming on the gel.
Huh.
Each band representing a group of DNA fragments of a specific size.
It's like those finish line photos where you can see who's ahead by a hair.
Yeah, I like that.
But how does this help us understand the disease?
Here's the key. You compare the banding patterns
Okay.
between the healthy individuals and those with the disease.
Right.
If there's a difference in the pattern,
Uh-huh.
say a band is missing,
Right.
or an extra band appears in the disease samples,
Okay.
that could point to a genetic variation linked to the disease.
Wow, so it's like a detective using fingerprints to solve a case.
Right.
Those subtle differences in the DNA,
Yeah.
revealed by the agarose gel,
Huh.
could hold the clues to understanding the disease.
Absolutely.
That's incredible.
It is, and this kind of analysis is the foundation for a lot of genetic research,
Wow.
from understanding inherited disorders to developing personalized medicine.
Okay, so agarose gels are helping us unlock the secrets of our genes.
They are.
What about polyacrylamide? What kind of mysteries is it helping us solve?
Well, remember that molecular waste station we talked about,
Uh-huh.
SDS page,
Right.
which uses polyacrylamide gels?
Yeah.
Plays a starring role in protein research.
Okay.
Let's say scientists are studying a complex process like cell signaling,
All right.
which involves a cascade of different proteins interacting with each other.
Okay, so it's like a relay race at the molecular level,
Right.
with each protein passing the baton to the next one.
Exactly. Now imagine you want to identify all the proteins involved in this relay race.
Oh.
You take a sample of cells engaged in the signaling process,
Right.
extract all the proteins,
Uh-huh.
and run them on an SDS page gel.
So each protein, stripped down to its basic size by the SDS races, through the polyacrylamide gel,
Right.
leaving behind those telltale bands based on their molecular weight.
Exactly. And this gives researchers a snapshot of all the protein players
Yeah.
involved in that particular cell signaling pathway.
Okay.
It's like taking a team photo of the molecular relay team.
I can see how powerful this technique is.
Yeah.
But knowing the molecular weights of these proteins, what can scientists actually do with that information?
That's where it gets really interesting. Knowing a protein's molecular weight can help identify it,
Okay.
like matching a fingerprint to a suspect. It can also provide clues about its structure and function.
All right.
Think of it this way. A smaller protein might be a nimble messenger,
Uh-huh.
while a larger protein might be a complex machine with multiple moving parts.
So SDS page using polyacrylamide isn't just about separating proteins.
Right.
It's about understanding their roles in the intricate dance of life.
Precisely. And this knowledge is fundamental to so many areas of biological and medical research, from developing new drugs to understanding how diseases progress at the molecular level.
This is all so fascinating.
Yeah, it is.
It really highlights how crucial it is for researchers to choose the right gel for their specific research question.
It is.
I mean, using the wrong gel would be like trying to run a marathon in flip-flops.
That's a good one.
You're just not going to get very far.
That's a great analogy. Choosing the right gel is crucial for getting accurate and meaningful results.
Right.
And it's not always a simple decision.
Okay.
There are several factors researchers need to consider beyond just the size of the molecules they're working with.
Okay, so it's not just about agarose for big molecules and polyacrylamide for small ones.
Right.
What other factors come into play?
Yeah.
So we've established that picking the right gel is like choosing the right running shoes for a race. What other factors, besides the size of the molecules, help scientists decide between agarose and polyacrylamide?
Resolution is a big one.
Okay.
If you need to separate molecules that are very close in size, polyacrylamide is the clear winner.
Right.
Think of it this way. Agarose is like using a standard ruler,
Okay.
while polyacrylamide is like using a micrometer.
Okay, so polyacrylamide lets you see those minute differences, those subtle variations that might be crucial for certain types of research.
Yeah.
Like, would this be important for that SNP analysis we touched on earlier?
Absolutely. SNP analysis, or single nucleotide polymorphism analysis, is all about identifying tiny variations in DNA sequences.
Okay.
Sometimes just a single letter difference in the genetic code.
Right.
These tiny changes can have big impacts,
Wow.
influencing everything from disease susceptibility to how individuals respond to medications.
So polyacrylamide, with its high resolving power, allows scientists to zoom in and pinpoint those single-letter variations. Those SNPs that might hold the key to understanding a disease or developing personalized treatments.
Right.
It's like finding a tiny typo in a massive book
Yeah.
that completely changes the meaning of a sentence.
Precisely. And it's not just SNPs.
Okay.
Polyacrylamide is also essential for techniques like protein sequencing, where researchers need to determine the exact order of amino acids in a protein.
Wow. Talk about getting into the nitty-gritty details.
Yeah.
So we've got size and resolution.
Right.
What other factors come into play when choosing a gel?
Well, practicality matters too.
Okay.
Agarose gels are relatively easy to prepare, like whipping up a batch of instant pudding.
Oh, nice.
Polyacrylamide gels, on the other hand, require a bit more finesse.
Okay.
They involve a chemical reaction, polymerization, to form the gel matrix,
Right.
which can be a bit more time-consuming and requires careful handling.
So agarose is the go-to for quick and straightforward separations.
Yeah.
While polyacrylamide is worth the extra effort when you need that higher resolution and precision. It's a trade-off, like choosing between convenience and specialized tools for a specific project.
Exactly. Ultimately, the choice of gel comes down to the research question and the level of detail needed.
Well, I have to say, this deep dive has been an eye-opener.
Oh, good.
I started out thinking about gels as these simple, almost mundane lab tools.
Right.
But now I see them as these powerful instruments that are helping scientists unravel the complexities of life itself.
It's remarkable, isn't it?
It is.
From separating DNA fragments to identifying proteins and even pinpointing single letter changes in our genetic code.
Yeah.
These gels are at the forefront of so many groundbreaking discoveries.
And it makes you wonder what's next.
For sure.
What new advancements in gel electrophoresis will emerge, and what new mysteries will they help us solve?
That's the beauty of science. It's a never-ending journey of discovery.
Absolutely.
Perhaps we'll see the development of even more advanced gel matrices,
Okay.
capable of even finer separations, revealing details we can only dream of today.
It's exciting to think about the possibilities.
It is.
Well, on that note, I think we've reached the end of our deep dive.
All right.
Thank you for joining us on this exploration of agarose and polyacrylamide gels.
Thanks for listening, everybody.
We hope you've gained a new appreciation for the power of these seemingly simple tools and the incredible insights they provide into the molecular world.
Absolutely.
And if this deep dive has sparked your curiosity, we encourage you to explore further.
Yeah.
The world of science is vast and full of wonders waiting to be discovered.
It is.
So keep learning, keep asking questions, and keep diving deep until next time.
See ya.